And now for the long-awaited follow-up to Evolution 2, Part 2. So in the first part, what did we really talk about? We talked about having homologous characteristics and what's so good about them and how do we use them to actually understand evolutionary history. Okay, so in this part of the video, what we're going to really look at is build on this idea of homologous characteristics and use and see what is the real evidence we have for evolution. Okay, so that's the whole idea, building on the topic of homologous characteristics and what really is the evidence for evolution. So when it comes to evolution, we really categorize the evidence that we get into two broad categories. One is that of homologies, and the other is the idea about where the organisms can be found, but we'll talk about that later. Okay, so on to the idea of homologies. We've actually talked about this previously. Okay, so what are homologies? Well, homologies are a fundamental similarity in organisms that is based on having common descent. Okay, so it's a fundamental similarity based on having common descent. In other words, it means that they have a certain feature that is probably inherited from a common ancestor. Okay, so we talked about this also in tutorial, the opposite being analogy. Okay, so homology, the opposite is analogy, where actually it comes from the idea of analogous structures, where you have two structures that are totally different, but they end up looking the same due to convergent evolution. So we are not going to use analogous structures, we are using homologous structures. So under homologies, we actually have two broad categories. The first being anatomical homologies, using anatomy, okay, various body parts. And on the other side, we've talked about this quite a bit in the other video, that is on molecular homology. Right, so and the other idea that's not actually related to homologies is that of biogeography, and we'll talk a bit about that towards the end of this video. So now let's focus on anatomical homology, and actually this also includes the idea of using fossil records, okay, because fossil records are also using anatomical homology, and you're comparing the dead and what is extinct to what is currently alive. Okay, so the whole basis of using anatomical homology is the very fact that we know that organisms, okay, we know that organisms with anatomical homology, they often have common morphological structures shared with a common ancestor. So the focus is really actually on morphological structures. It's not meaning to say that there are exactly the same structures, but rather like the limbs, and or you might say the head structure or something like that. Okay, so for example, there can be bones, organs, and even related growth structural features. Okay, there are certain bones, organs that may be conserved but looking very different, and these characteristics, they are often altered by natural selection in the descendants. So there can be a very early structure, but over time this was altered by natural selection. Okay, so all of this together, anatomical homology, it aids in the measuring of your degree of relatedness. Okay, so by using anatomical homology, you can actually estimate the degree of relatedness. But remember what we talked about earlier, it is not without any problems. Anatomical homology, if you purely rely on anatomical appearance, we talked about this, it is not going to be purely a very reliable indicator of relatedness. It might show you some connections, but there will not be a very reliable indicator of relatedness. So when it comes to talking about anatomical homology, there are a lot of good examples out there, and there are a few that are very commonly used. Okay, so one of example is the idea of having vestigial structures. There are many different vestigial structures in a lot of organisms. Okay, in one good example in humans is the tailbone. Okay, the tailbone serves almost no purpose, but it's still preserved, and you can compare it to apes and other monkeys that still have a tail. Okay, so there are structures that have been preserved over time and through evolution, but now they serve no purpose. Okay, another good example that is commonly used is like the pelvic bone in whales. Okay, which came from the hind foot in a land-based mammal and eventually just regressed and now serves no purpose. One very precise category of anatomical homologies that are commonly talked about is that of the pentadactyl limbs of tetrapods, okay, or vertebrates that have 4 limbs. So how are pentadactyl limbs actually homologous? And how do we compare in the limbs, right? How? How do you do that? So when it comes to pentadactyl limbs, we talk about the idea of the 4 limbs. That means the front limbs, okay? The maybe in humans the hands, or in other animals the forelimbs. Okay, in different related species, they end up having the same arrangement of bones, but they have very different functions, and they also end up looking superficially different. That means on the outside they look very different. So even amongst mammals, you will see this great drastic different adaptations and the functions, and looking very superficially different in the limbs itself. One example is that of cats versus whales, right? So cats have their limbs adapted to walking, but whales have adapted it to swimming. But if you actually look at the bone structure, they actually look quite similar, and because of that you can actually use this to analyze the relationship and how they have evolved from a very early mammal. And then remember when we started talking about evolution, we said that the whole idea of evolution actually boils down to the idea that there is descent with modification. Okay, the key phrase, descent with modification. And how does this anatomical homology support it? Well, okay, having 5-digit pentadactyl limb structure, it is actually going to be altered by natural selection. So all these animals, they have this overall limb structure, but it is altered by natural selection. So what is that alteration? Is really that modification ends up suiting each species and their specialized functions or environments. And this really shows us that there is evolution and there is natural selection, because there is a variation of the pentadactyl limb structure that is a product of the whole process of natural selection. Okay, with that, I'm actually done talking about anatomical homology. And if you're ready, let's move on to molecular homology. Now, we've talked about molecular homology at quite great lengths in the previous video. Okay, so this is just a quick summary of all the main points. And the basis of molecular homology, again, it boils down to us using the genetic code. And this genetic code is basically your DNA. It is universal. It is shared amongst all organisms, be it humans, be it beetles, insects, amphibians, right? So organisms that share common ancestors, we know that they will have quite similar or very similar DNA, RNA, and amino acid sequences, right? So apart from having the genetic code being universal, we actually have seen the idea that homologous genes, right? Homologous genes, they share significant sequence homology. Homologous genes share significant sequence homology. Certain sequences are conserved, and certain sequences, they can mutate, they can change quite easily. So homologous genes, when they are expressed, they will tend to produce proteins with same or very closely related and similar functions. And this boils down again to the idea, the fact that organisms that share common ancestors, they will have similar DNA. And from there, you can express proteins that have same or similar functions. And again, when we use molecular homology, it can aid in the measuring of the degree of relatedness amongst various organisms. So molecular homology can be used to aid in the measuring of the degree of relatedness. And you know that the greater the sequence similarity between homologous genes, the greater the sequence similarity between homologous genes, the more closely related the two species are. So now when we actually have homologous genes and we can actually use them to compare, right? This also then supports the idea of evolution because it supports having descent with modification. If you have homologous genes that can share significant sequence homology, it supports the idea of having descent with modification, right? And actually this is because, as nucleotide sequences in ancestral genes, they become modified due to accumulation of mutations. So there is that modification, the idea of modification, right? Ancestral genes were modified. On top of that, right, in the ancestor species, in the ancestor organisms, in the environment that they lived, there would have been many different selection pressures due to the different environments, like the predators and the environment that they lived in. So we say that different organisms would have had different selection pressures, and this actually supports the idea of descent with modification, and we've seen this in evolution 1. So overall, some mutations ended up being favored over others and they were preserved. And as a result, this could then be inherited by the next offspring and carried forward. And this again boils down to the core idea of evolution. Now, I'll leave you to go and read up about the examples, but we've already covered about why certain genes are used in the tutorial, right? So there are a lot of examples that are used for molecular homology, many different genes. So they are commonly used genes. Okay, so they are found throughout the DNA, but certain genes like the cytochrome B and C genes and the P53 genes, they end up being very good genes that can be used to compare between different organisms. And I'll leave you to go and read up about why certain genes are used and why some other genes may not be used. Okay, and with that, we're actually done talking about homologies. So let's move on to biogeography. And yes, I know we've talked about bio mixing with chemistry, bio mixing with maths, now bio mixes with geography. So really it is the study of the past and present geographical distribution of organisms using their ranges and where they may be found. So you use the past and present geographical distribution of organisms. And why we can use this is because we know, and through a lot of studies we have found that it follows patterns of evolution and movement of tectonic plates over geological time. So we are really talking about the geographical distribution of organisms. It really follows patterns of movement of the tectonic plates. And all these movements, they are actually shaped by natural selection. Okay, and the changes, they are also shaped by natural selection due to the differences in local environments. Okay, there may be new habitats or challenges in the current habitats. So we'll look at certain key ideas that is linked to biogeography. The first key idea, and this is brought about by a lot of studies and a lot of fossil studies, and we know that closely related species and common ancestors, they should be present in the same geographical region. So in the same geographical region, you should be able to find closely related species. And there are many examples for this, like elephants and whatsoever. But one great example is that of great apes, and they're only found in Africa and Asia, which through a lot of geographical studies, we know that actually they were all connected. But because of this, you would not be able to find great apes in Australia and the Americas. So the second idea boils down to that of continental drift, and it's actually quite linked to this previous idea that we talked about having same geographical region. So continental drift is the idea, or is the theory, that if related species are not distributed in the same geographical region, they tend to be older, okay, and actually continental drift can explain it. So their common ancestors used to live on maybe the supercontinent. And this is really seen in fossils that are found on totally different regions. What do I mean by totally different regions? Like Africa and South America. Okay, they're separated so long ago, but once they were on the supercontinent. Okay, so continental drift also actually explains if there's a problem if you don't find closely related species on the same geographical region. Then the third and actually final key idea that we are looking at with regards to biogeography is that of island biogeography. Why there are certain distributions on islands as opposed to the mainlands. Okay, so island biogeography talks about the biogeographic distribution, and in a lot of cases we've seen the biogeographic distribution of finches on the Galapagos Islands. It supports evolutionary deduction that there was descent with modification. So it is this biogeographic distribution on an island that supports the idea of descent with modification, because we know now that island species evolved from ancestral mainland species, and this is commonly seen in a lot of islands. And this happens because there is adaptive radiation as a result of natural selection. So remember, there's a very good example in the Galapagos finches on the islands versus the mainland finch. Okay, so this is island biogeography. And with that, actually, we're done looking at the evidences for evolution. Thank you very much, and I will see you around.